What we have seen thus far today is that psychiatric and neurological diseases are genetically complex illnesses. They often have an interaction with the environment, and because of their location in the brain, they are incredibly difficult to study on the level of their molecular genetics. Alzheimer's disease is also a neurological illness, but it is also an illness of aging, where we see a doubling of the prevalence of Alzheimer's for every 5 years of age above 65 in a population. What we see here is the outcome of a genome-wide association study for late onset Alzheimer's disease. As we saw in the previous lecture, early onset Alzheimer's disease is typically a Mendelian condition, and it involves mutations in the genes APP or PSEN1 or PSEN2. This results in differences in the cleavage of the APP protein and increased deposition of the A-beta 42 protein, increasing the amounts of senile plaques in the brain. These senile plaques are toxic, leading to neuronal death and eventually shrinkage of the brain. What we see here then, as I've said, is late onset Alzheimer's disease. In this case, there is the absence of these very high penetrance mutations, and we are back in the environment of multiple common and rare alleles, each of which has very low odds ratios or relative risk in the context of the phenotype. So let's see what we have here. First of all, there are multiple different genes that we can see. We're not going to go into the names of all of these genes today or any of them, but it's suffice to say that like other illnesses that we've studied, there are multiple loci, each of which only has a very low odds ratio, with the exception of ApoE. The proportion of patients who carry the risk ApoE allele can explain up to 10% in the variance of age and the onset of dementia. In other words, depending on the age of onset of Alzheimer's, you can explain up to 10% of that variation in a population by the presence or absence of a particular allele of the ApoE gene. The particular allele, which is the ApoE4 allele, also has very high and variable odds ratios, ranging in some studies from as low as 3 to much higher than 11. In other words, this is a very heterogeneous effect on phenotype. And there is a lot of discussion as to why this is, ranging from the existence of endophenotypes. In other words, there are different phenotypes that we would commonly call Alzheimer's disease, through to, of course, the genetic makeup of the individual and how that genetic background influences the likelihood that this ApoE4 allele will lead to late onset Alzheimer's disease. However, with the exception of this ApoE4 allele in the ApoE gene, most or all other SNPs have odds ratios of around about 1.1 to 1.3. There are 3 common alleles, of which ApoE3 is the most common, and it's found at a frequency of 0.8 in Europeans. 25% of Europeans are heterozygous for ApoE4, with a 5-fold increased risk of late onset Alzheimer's disease. The 2% of the population who are ApoE4 homozygotes, however, they have a 10-fold increased risk of developing late onset Alzheimer's disease. Now, remember, this is a risk relative to the healthy population. So having the ApoE4 allele, although it is a significant risk, does not ensure that that person will develop the disease. And again, this is a common theme that we keep coming back to in these genetically complex diseases, that even with very high odds ratios, which is certainly can be the case with ApoE4, it does not guarantee or ensure that this person will become ill as a result of this particular allele. So at least 1/3 of all people with late onset Alzheimer's disease who do not carry the ApoE4 risk allele will develop Alzheimer's, and not all ApoE4 carriers will develop late onset Alzheimer's disease. So again, we're looking at a disease which we have a very high risk allele, but it by itself does not explain or is sufficient for the onset of this particular disease. So the major genetic risk factor for late onset Alzheimer's disease, as I've said, is the allele ApoE4. And it was first identified through linkage studies, which are a type of family genetic association study. It is known that ApoE4 interacts with A-beta in the amyloid plaques, indicating a direct association between the protein ApoE and A-beta in Alzheimer's disease pathogenesis. As I've said, not all people with the ApoE4 allele will develop late onset Alzheimer's disease, and not all people who have the E4 allele will develop the disease either. So what we see is that we have the ApoE here, which is thought to be important for the clearance of this A-beta peptide from the brain, and it is believed that the ApoE4 allele in some way interferes with this process of the ApoE gene. So illness then is correlated with altered patterns of gene expression. Changes in gene expression has a genetic and environmental basis. If we think about the ApoE gene, it has multiple alleles. ApoE2 is mildly protective, whereas ApoE4 is a significant risk factor for late onset Alzheimer's disease. Healthy individuals with at least one ApoE4 allele share a pattern of altered gene expression with individuals not carrying the ApoE4 allele, but who have late onset Alzheimer's disease. So let's see, just take a minute to just say that again. So what happened is that there was a study done, and again, this was done on post-mortem brains, where the researchers took brains of individuals who were heterozygotes for ApoE4. These individuals, although they were carriers for the risk allele ApoE4, did not have Alzheimer's disease. Their brains were compared with the brains of individuals who did not carry any of the E4 alleles. In other words, they either had E2 or E3 alleles, and there was a gene expression comparison done between them. What it was found is that the ApoE4 allele itself started to initiate changes in gene expression, which were in part similar to those brains that were already showing symptoms of Alzheimer's. In other words, if you like, the ApoE4 allele seemed to be initiating a pre-symptomatic change in the gene expression pattern in the brains of carriers of this particular allele. And that's what's shown in this diagram here. So what we see is we see increasing risk of late onset Alzheimer's disease. If we start here at the white or yellow color, there is no risk, through to a risk of 1, in which case you have the disease. So for example, here we have ApoE3 carriers who do have late onset Alzheimer's disease. They're coded as red because they have the disease. Here we have healthy ApoE4 carriers. In other words, they don't have Alzheimer's, but they do carry an allele, and they are classed somewhere intermediate in this region here, not having the disease, but exhibiting changes in terms of gene expression that would put them on the high end of risk of developing late onset Alzheimer's disease. We can see that more clearly in this diagram here. So what we can see is that by having the allele ApoE4, and by looking at patterns of gene expression in the brains of these healthy individuals carrying this allele, they share some genes with individuals who are already showing symptoms of late onset Alzheimer's disease. Now, what's interesting is that as we age, and particularly people who are over 85, they also show changes in gene expression patterns in their brain, which are similar to late onset Alzheimer's disease. So in other words, we're seeing the brains of individuals with late onset Alzheimer's disease, they show patterns, changes in patterns of gene expression, which are firstly correlated with the aging process, and secondly correlated with the actual pathology of the disease. And it is here that they intersect with carriers of the ApoE4 allele. What's interesting is is that the genes in this part of the graph here that are common to people who have late onset Alzheimer's disease and to the healthy carriers of the risk allele, is that these genes show altered expression in genes that are involved in the trafficking of the APP protein and its processing. So in other words, there seems to be a correlation between disruption of this pathway that processes and clears these A-beta peptides from the brain. ApoE then is important for A-beta clearance from the brain, and it is thought that the ApoE4 allele does this less efficiently. If we look at the brains of individuals who are carrying the ApoE4 allele, we see changes in patterns of gene expression, and those genes which show changes in expression tend to be on pathways involved in the clearance and processing of the A-beta peptides. So in summary then, Alzheimer's disease is a neurodegenerative disease. In common with other neurological illnesses, molecular studies of brain tissue can only be done post-mortem, which raises the question then, how might we study the effect of AD-associated mutations and polymorphisms in human neurons?